Hey, Marin Academy students and teachers, welcome to the Cloning Podcast. My name is Addie Winship and I am a senior, and in our time together today, we will cover some common misconceptions about cloning and learn some new facts about this compelling branch of genetics. You might be wondering, how does cloning work, who is the famous Dolly, and what is happening in the cloning and genetics community in the greater Bay Area and even local Marin County. Without further ado, let's dive deeper into these questions. To start out, we will travel back in time to the late 1800s, where scientist Hans Dirch was on the brink of an amazing scientific breakthrough. Cloning has been around since the beginning of the time, with self-replicating bacteria, plants, and even animals existing long before humans even knew what cloning was. Hans Adolf Eduard Dirch was a German biologist and philosopher hungry for answers. He studied hard in a lab in Naples, Italy, studying sea urchin embryos. One day, he found that when he separated a newly fertilized embryo in half, each divided cell developed into a completely new sea urchin. Whoa. He had created a clone. After that, a watershed of genetic work followed in pursuit. How was Dirch able to do this? How did he simply split the cells in half, proceeding to grow two new, completely different, but genetically the same organisms? This all lies in the science behind cloning. There are two main techniques to cloning organisms that have been developed over the past century, which include the splitting of embryos and somatic cell nuclear transfer. The splitting of embryos is complex, but once understood, it actually can be quite simple. By definition, an embryo is an unhatched or developing offspring. And after the first day of fertilization, when the egg and sperm combine, an embryo begins to grow by the splitting of cells, which is known as the process of mitosis. After 8 weeks, it becomes what we would know as a fetus in the terms of human development. Now, the first way to go about splitting embryos is through blastomere biopsy separation. That is a mouthful. Blastomere biopsy separation is when the first cells in an embryo split. This phase of development is called a blastomere cell phase. A couple of the blastomere cells are removed from the zona pellucida, which is the young embryo outside shell, and moved into an empty zona pellucida, where they develop and form into a new organism. The other way to undergo the splitting of embryos is bisection in the blastocyst embryo. To put this in less fancy terms, this means cutting an embryo that consists of only 6 to 32 cells directly in half, and from there they grow separately, which is the technique that Dirch used on the sea urchin. Okay, deep breaths everybody. That was a lot. Still, it is only the first way to undergo the cloning process. Let's dive deeper into the second. The second main way to clone an organism is somatic cell nuclear transfer. Somatic cell nuclear transfer is the process of taking an egg from a female organism and inserting the DNA of a regular cell from another organism, also known as somatic cell nuclear transfer. This happens by denuclearizing the egg cell, which is the process of removing the nucleus and all of its DNA from the egg cell so that it becomes a sac to hold the DNA from the regular cell. Then the DNA from the somatic cell, or regular cell, is extracted and inserted into the egg through cell fusion. This artificially inseminated egg is inserted into a surrogate mother's uterus, where it grows and is born into the clone of the parent that donated its somatic cell or regular cell DNA. Enough with the sciencey details. Somatic cell nuclear transfer is a popular way to undergo cloning many animals. A prime example of this is Dolly. If you've been paying attention, I mentioned something about Dolly earlier in the podcast. Who is Dolly? Dolly was not a cat, not a dog, not a duck, not a human. But I'll give you a hint first. Hopefully you guessed that Dolly was a sheep. She was also the first mammal successfully cloned from an adult cell, or from what we learned, a somatic cell. Let's travel back in time to the 1990s to learn how Dolly was made. Far away in Edinburgh, Scotland, under grey clouds and surrounded by old architecture, Professor Keith Campbell was concocting something. At the Roslin Institute, science removed a cell from an udder gland of a 6-year-old Finn Dorset sheep, then proceeded to extract the DNA from the cell and fuse it with an egg that has had its nucleus removed. The egg was taken from a Scottish black-faced sheep. This might sound familiar, and you guessed it, it's somatic cell nuclear transfer. The egg developed in a lab until it began to divide and grow more cells, which is the process of mitosis again, before it was implanted into the uterus of a mother sheep. A crazy fact about this is that they attempted this process 227 times, and only 29 embryos were successfully created. And even then, they only had one success, which was Dolly. After many trials and months later, Dolly was born and was an exact replica of the Finn Dorset sheep who donated the udder gland. Dolly went on to live a normal life and had 6 lambs, and she sadly was diagnosed with arthritis and died when she was 6.5 from tumor growth. R.I.P. Dolly. Okay, so now we know how people clone things, but why do they? People don't just clone animals for the sake of cloning as they did for Dolly. There are other reasons why people clone organisms. There are 3 main reasons why cloning exists. The first is gene cloning, which is the copying of segments of genes or DNA. This makes new copies of DNA and can be used in the medical field for vaccines and medicine and so much more. The second is reproductive cloning, which is the cloning of whole organisms or animals. This has yet to be explored more, but can be a very powerful tool if used correctly. For example, maybe we can clone humans in the future. The last use of cloning is therapeutic cloning. This is when the newer formed embryo cells are used to create tissues to replace broken and diseased ones. This is still developing and could be a massive science medicine breakthrough in the future. Needless to say, there is still a lot more to know about cloning, and even though it feels like you might not interact with it in your life, you just might. In our local Marin County, there are major cloning operations that have happened in the past and even are happening present. BioArts International was a company startup founded in 2008 by a man named Lou Hawthorne. Hawthorne himself had kids who attended Marin Academy not so long ago. This company had a license for somatic cell nuclear transfer, which was the same cloning technique that was used to create Dolly the sheep. This company would create and clone beloved dogs for a very high price. You yourself could have cloned your dog for a high price. In order to publicize his company, in 2008, he made the Golden Clone Giveaway contest, in which one lucky dog lover would be selected to have his or her favorite pet duplicated from DNA. Participants had to write a 500-word essay about why their dog should be replicated, and the winner would win a live replication of their pet. I think this is just crazy. Would you participate in this contest? Needless to say, cloning is a very interesting branch of genetics that is continuously evolving. Get out into the scientific community and stay curious, and don't forget to look for contest postings to replicate household pets. Thank you for listening, and I'll see you soon.